introduce our next expert to talk today, Dr. Greg Michelotti. So Greg is the Director of International Population Health. Greg got his Ph.D. at the University of South Carolina. And before he joined Metabolome, which is a couple of years ago, but he never tells anyone that, he led a translational research laboratory at Duke University, where he did work on incorporating multi-omics approaches to understand the molecular basis of liver disease and NASH progression. Since joining Metabolome, Greg leads biology efforts to identify metabolic signatures of disease in large human population cohorts. And today, Greg will talk about how our metabolomics platform can be used to understand the human outcomes. So, thank you, Greg. The floor is yours.
Thank you, Simone. Appreciate that introduction. And I don't know if my colleagues know this, but I actually was a client before I joined Metabolon because in my work at Duke, where we were studying fibrogenic signaling in liver disease, we had been doing genomic and transcriptomic approaches for many years, and it wasn't really elucidating the pathways that we needed to get to understand liver progression, liver disease. And it was a project that I did with Metabolon that took us down really novel and exciting pathways down one-carbon metabolism that led me to see, as a client, how exciting metabolomics could be. And I've since joined them to evangelize on what I believe and know to be the power of metabolomics to really deconvolute complex disease signals to understand human health. A lot of what I talk about, there's going to be some redundancy in the first part of the talk, but I'm going to put a little more context behind what Brian talked about in his review within the context of human health. But because he's the elder statesman and has been at Metabolon longer than I have, he got to steal my thunder a bit on some of the points I wanted to make. So, as Brian said, Metabolon is a leader in global metabolomics provision, and metabolomics tends to be thought of as the next omics coming in after genomics. And I would like to challenge your thinking on that, to think about metabolomics more as a founding omics, because we're resting on over 100 years of metabolic research, from the pioneering work of Otto Warburg to understand glycolytic signaling in cancer biology, to all the work that came after that to map metabolic pathways from enzymes so we understand how they're catabolized, how they're generated, how they interact with each other. So from this work that happened over 100 years ago, we have a very deep understanding of the biochemical pathways, their ontology, and their annotation. And we focused the last 20 years on developing the best technology we can to comprehensively profile these by developing unrivaled capabilities to get massive coverage to be able to provide biological insights into samples that ultimately, as Brian talked about, and I'll talk about a little bit, have clinical applications. And the one thing I want to get out, besides of an overview of what metabolism is, because I think we all know it's a conversion of food to energy that drives life, is that any transition from a homeostatic state to a disease state requires a change in a biochemical step. So in that view, every disease is a metabolic disease. Metabolism can be used across all biological questions and all processes. And importantly, a reversion of that disease state back to a homeostatic state requires the same change or identification of a change in a biochemical step. And that uniquely poises metabolomics as a tool to be able to be applicable across all disease states. And so in that view, it really has powerful applications in both diagnostic, prognostic, and therapeutic applications. And as Brian said, metabolomics is unique in that it integrates all the information from the upstream omics. So that flow of genetic information, the central dogma, molecular biology, genomics to proteomics to metabolites, has a proximity to a phenotype where metabolomics really represents a definitive representation of a phenotype. And while genomics and proteomics are powerful, not disputing that, they suffer from limitations that really preclude our ability to understand their functional impact in deconvoluting complex disease signals. Genomics, for example, suffers from a lack of knowledge of gene function. We still understand the function of a really small number of genes relative to all the expressed genes out there. And then you start talking about microRNAs and non-expressed genes. It really confounds our ability to understand the function of genes. So we have a poor understanding of the gene ontology, what their function is, and how they interact with each other. Going down the line to proteomics, it suffers from a distinct set of complexities in that the proteomic technologies look at the levels of proteins, but the levels don't tell you the activities per se. And the activities of these proteins are influenced by a bewildering array of modifications, including over 300 post-translational modifications, cofactor binding, typically by small molecules, alternate splicing, subcellular localization, and all these factors influence our ability or confound our ability to understand the function of the proteins that largely eludes just looking at the levels of these proteins. Metabolomics, as Brian said, though, get around this because we have a deep understanding of the functional annotations from resting over 100 years, as I said, on metabolomics research, so we know how these molecules interact, and it gives us the ability to be able to interpret the changes that we see in biological systems. And we talked about the translatability question, that the metabolites are functionally and structurally conserved throughout the phylogenetic spectrum. So as Brian said, citrate in plants, in bacteria, these are all translatable across the phylogenetic spectrum, where the other upstream omics tend to be more confounded in that way because different genes, different proteins can have different functions in different model systems. And just really briefly, because Brian touched on this, metabolites and metabolomics integrates both genetic and non-genetic information to provide a comprehensive view of the phenotype that we know about the information that incorporates from the upstream omics, genes and proteins, but also the exogenous factors that come from microbiome and lifestyle. And as Brian talked about, microbiome and lifestyle and exogenous influences from the environment have a profound impact, excuse me, on health outcomes. And those signals or inputs are largely missed, specifically in genetic approaches that don't incorporate those into understanding human health outcomes. And nowhere is this perhaps more relevant or salient than in looking at cancer biology. And I bring cancer up specifically because it's thought to be the quintessential genetic disease. But when you start to think about all the genetic drivers of disease, and I looked at c-Myc, for example. So I studied c-Myc as a postdoc, and I think we're 45 years into looking at proto-oncogene function, and we're no closer now to understanding how c-Myc promotes cancer than we were 45 years ago, despite consuming the lives and structural and scientific careers of a huge number of scientists. We still don't understand the function of these things and these pathways. So these genetic drivers, while they're potent drivers of oncogenesis, they all have powerful metabolic underpinnings. So when you start to break down the hallmarks of cancer and understand what the hallmarks of cancers are, and this is an adaptation from Robert Weinberg's proposal of what the hallmarks of cancer are, each of those processes, whether it's immune invasion or deregulated cellular proliferation or apoptotic signaling, they're all driven or underpinned by metabolic pathways. So as I said, every disease is essentially a metabolic disease because any one of these transitions requires distinct changes in metabolic pathways that is captured in a comprehensive metabolomic profiling approach. And to really nail the point, health is not all in your genes because genes assess the risk of moving into a disease, whereas metabolomics is the proximity to the phenotype to the definitive representation of those outcomes. And you can take genetically identical models, and this is a preclinical model. For example, black 6 mice are genetically identical. And you can offer them 2 different diets or create a study with 2 different diets. And twins, these studies have also been recapitulated in humans in terms of specific twins, that you can give genetic identity models different diets, an unrestricted high caloric or high fat diet versus a caloric restriction diet. And with genetic identity, you get very different changes in health outcomes. On one hand, with overnutrition, you get disease and early death. And with caloric restriction or controlling the metabolism or metabolic inputs, you get a long and healthy life. And of course, this is subject to intense investigation looking at the pathways that are supporting this to help promote salubrious and long-term health. And this is in large part, again, microbiome and microbially influenced. And Brian talked about some of the interplay between host genetics and microbial composition in the gut and in other communities in the bodies, and oral and pulmonary. There are other microbiome communities, microbial communities. That a definitive study done by Ron Segal in Israel, the Weizmann Institute, looked at repatriation of the Israeli diaspora back into the Israeli state back in the 50s. And the question was, looking at a relatively homogeneous genetic population of Israelis that repatriated back to Israel, what is the gut microbiome composition in this population? And they found that environment has a much, much more powerful role in shaping gut microbial composition relative to host genetics. So all these things reflect distinct signals and processes from host genetics that come from and can be captured from metabolic identification or profiling. And Brian talked about the panels that we do to support, because the goal is to capture as much biology as we can from all these different pathways. Brian talked about the global panel for discovery applications, and he talked about biology-focused assays that we also support from the complex lipid panels that he talked about. And one that I'm really excited about is the inflammatory mediated panel that looks at, even though we look for those on the global panel in terms of prostaglandins and leukotrienes, which we detect a very small number of them on the global panel, the pro-resolving mediators that have come into a lot of focus lately that help resolve inflammation, those lipid mediators like resolvins and protectins and mucins and sulfatides, while sulfatides are being developed. They have been put into a specific targeted panel now because we largely missed that biology on the global panel, but we have a specific targeted panel to capture inflammatory mediators to understand who promotes, in what situations inflammation is promoted, and in what situations it can be resolved. And this has relevance, like Brian said, across the entire spectrum from basic research to discovery to clinical development as well. So I won't belabor that point. So we really do view ourselves as a leader in metabolomic profiling, as partially evidenced by all the publications that have come out from Metabolomics, and specifically using our technology. We have over 3,000 peer-reviewed publications now that have reported on our data, a large portion of which are in high-impact journals, and a number of which we are co-authors on because we have a specific scientific and intellectual investment in our data and in this work. So we are co-authors on a large number of papers as well because we're involved in this process. And this next part of the talk, I'm going to talk about some of these specific applications of metabolomic profiling, how investigators are using our data in the health outcome space based on publications that we've put out recently. And I'm only going to put 6 up because Brian covered a huge number of areas that are largely redundant because of microbial influences and all that. So I don't want to convey death by case study here. So I'll represent some high-level views of this and happy to talk about, because keep in mind, this is just a subset of everything that we've done. And just keep in mind, because every disease is a metabolic disease, if there is a disease of interest or a topic to discuss, chances are we've done something on that and we'll be able to comment on that. The first one I want to talk about is a discovery application of the global panel. And these largely reflect plasma profiling. So in large population studies, the most attractive sample type to get is either plasma or serum. Urine reflects another popular matrix, but it's not homeostatically maintained the way plasma or serum are. So a lot of the things I'll talk about are in the focus of plasma and serum and capturing signals in the circulation to understand complex disease. So working with Claudia Langenberg at Queen's Mary University in London, she asked a question from her EPIC cohort, so profiling 12,000 EPIC Norfolk patients or subjects to understand metabolic signatures of simultaneously existing chronic disease conditions called multimorbidities. So 25% of subjects, now patients, are presenting at clinics with multiple chronic conditions. And it's reflected a challenge or created a challenge in the health community to treat, how do you treat someone who's presenting with multiple chronic conditions and which ones do you treat? The focus of the study was to understand what the metabolic signatures are for distinct chronic conditions and an aggregate for people with simultaneously multimorbidities, and to understand those signatures. So on the left there, an odds ratio of 95% confidence intervals from logistic regression analysis was performed, and then the metabolite disease associations were ordered by strength of association and directionality. And what emerges from that graph on the left is that when you zoom in, as we're zooming in here for the purpose of making the point, on what the metabolites are that are strongly associated, meaning they are associated with poor outcomes, or negatively inversely associated with better outcomes, that on the right, the negatively associated or associated with poor outcomes tend to be enriched in glycerophospholipids. And then when you look at the category on the right in blue, that these metabolites tend to be focused or enriched on microbially derived compounds. And it starts to identify what classes of metabolites, what pathways, what signatures are associated with poor outcomes. Could they be drivers? Could they be diagnostic? And which ones could have good outcomes? And one of the interesting things that emerged from the study is that 2/3 of the associated metabolites detected in the study, 2/3 were associated with multiple chronic conditions, whereas only 1/3 was associated with one chronic condition alone. And what that means is that there are shared etiologies to multiple chronic conditions that perhaps suggest that there could be opportunities for interventions, or at least diagnostics, to identify who's at risk for multiple chronic disease conditions. You can improve all chronic disease conditions if you can identify a shared metabolic perturbation and improve the lives really quickly in a really sad cohort with poor outcomes that have really significant health challenges. And then you also have the opportunity from the metabolites that are significant or specific for one multimorbidity or one disease only, to have therapeutic opportunities for those as well, diagnostic and therapeutic opportunities as well. And when you expand the aperture on the right and look across all the multimorbidities, and this is looking at a mirrored Manhattan plot, looking at the p-values. These are all, of course, surviving Bonferroni correction or multiple testing, that the metabolites on the top half of that graph, that image, are positively associated with those multimorbidities across all the diseases. And on the bottom part, it's the negative associations or inverse associations. And I know it's more of an eye chart, but I want you to kind of get the scatter of all the lines across all the diseases, that there are very potent signals. There's very potent metabolic perturbations that are happening in these disease states that are both positively associated with poor outcomes and inversely associated with poor outcomes. Interesting, cancers in the middle tended to be a little more quiet, with the exception of lung and colon cancers. And this tends to be because these were not well represented in the study. So it was a power question for the cancers, that in this specific cohort, there just weren't a lot of the specific cancer types that drove a high power to be able to identify this analysis. But I think it's an area for fertile investigation. A second approach is of metabolomics, how investigators have used our technology, is more of a multi-omics approach, to use metabolomics to functionally map variants of unknown significance in genetic studies. And I think this is arguably one of the most innovative applications of our technology, because the question is, or the idea is, that you can take your GWAS in a large population study, and when I say large population, I'm talking like genetic numbers. Like these are thousands of subjects, thousands of patients or subjects in the cohort. And the question is, can you identify a variant that results in a change or associates with a change in a biochemical step? And in that way, you're able to map the function of what that variant is doing in that individual. And if you'd asked me before the study was done if that would actually work, given some of the power of the study and the kind of powers that you need to see for allele frequencies for some of these, I would have said it would never have worked. It's a good thing that I wasn't listened to or I wasn't questioned, because it actually worked extremely well, and it's a very powerful approach now and well adopted. And the initial approach was discovered or vetted by Carsten Surrey at Weill Cornell. And in this specific example that I show here on the left, SLCO1B1 is a solute transporter where variants were known to be associated with increased mortality and increased risk of heart failure. But the mechanism was completely unknown. And it was a study that he did where he looked at about 6,000 subjects across two different cohorts, Twins UK, which Brian mentioned earlier, and then a Cora cohort, which is a German-specific cohort that acted as kind of a discovery and validation arm in the study. And he identified variants in the solute transporter that associated with levels of a single nucleotide or single metabolite called hexadecane-dioate, a dicarboxylic fatty acid that's typically involved in peroxisomal function. And that was a massive surprise because solute transporter was not thought to be relevant for this class of molecules, but clearly there's an association where this variant was associated with altered circulating levels in the plasma of this metabolite for individuals who had variants in that gene. And then validation came from another cohort called the ERIC cohort, led by Eric Boerwinkle at University of Texas Southwestern, where in that cohort, they identified a dosing effect of loss of function of alleles at a specific locus or specific allele in that same transporter, where you see that the putative loss of function homozygous allele results in a profound increase in circulating levels of the same metabolite in a distinct cohort. And because this is known to be associated with heart failure, the hypothesis is that this could be directly related, this metabolite could be directly related to the function of this variant that's known to be associated with increased risk of heart failure. So Christina Meni at King's College in London tested that hypothesis, and she actually dosed rats with this specific molecule. So starting at day 9 after an acclimation period, relative to placebo or vehicle treatment group, animals dosed with hexadecane-dioate showed a marked increase in systolic blood pressure, showing a specific and causal effect of this, that this could be the actual mechanism by which SLCO1B1 is mediating increased mortality and increased risk of heart failure. And that's because, according to World Health Organization statistics, as of 2020, there are 1.5 billion people in the world with hypertension, and 1 billion of them, 2/3, almost 1 billion people, are not receiving their targeted therapeutic therapy, and there's a lockstep association between increased blood pressure and increased mortality. So this study shows that perhaps there could be another way for new modalities and new treatments for disease which are desperately treated, because it remains a grave global concern. Our technology has also been used for biomarker assessment of target engagement, so more of a clinical application of this. This comes from a NASH area, so this is Rohit Lumba's work at UCSD, University of California, San Diego, where non-alcoholic steatohepatitis suffers from or results from dysregulation of de novo lipidogenesis and decreased fatty acid oxidation. So the thinking is that liver fat is one of the steps that go on to pathological sequelae that relate to non-alcoholic steatohepatitis or NASH or decompensated liver function. So the idea is if you can block that liver fat from ever being produced, you can improve NASH and be able to treat NASH. So they focused on a specific target, acetyl-CoA carboxylase, to which is a key enzyme that is involved or drives de novo lipogenesis. And it's known that there are some key markers of this enzyme that are malonate, malonyl carnitine, and beta-hydroxy beta-carnitine, and then butyryl carnitine that are known to be regulated or influenced by the function of this ACC enzyme. And so the first thing they did is characterize in a NASH population versus healthy controls, what do the levels change? Does the function of this enzyme correlate with what we'd expect from a disease profile? And you can see that 3 of these metabolites go up, whereas I think it's butyryl carnitine that doesn't change, but malonate, malonyl carnitine, and beta-hydroxy butyryl carnitine do increase in NASH relative to a healthy population, suggesting that this represents a good target to go after in terms of treating NASH. So we worked with them to do metabolic profiling on a specific panel of these metabolites to create a biomarker index from these, which is actually a ratio of malonate, malonyl carnitine over beta-hydroxybutyryl carnitine. And using this index that we're able to profile for them in this study, they're looking at healthy controls that were subjected to de novo lipogenesis. So these healthy control cohort was given labeled fructose to drive de novo hepatic lipogenesis, and the C13 incorporation into VLDLs was used to assess de novo lipogenesis. And in this healthy cohort, individuals were characterized this biochemical index or metabolic index from these metabolites, showed a strong correlation of increase in these metabolites versus de novo lipogenesis, the percent change in de novo lipogenesis, showing that this would be a good biomarker index to look at target availability and target engagement. And in the same population in a crossover study, they were either given placebo or the drug down here with this de novo lipogenesis that was tracked. And relative to the placebo group, you can see a marked decrease in the biomarker index for this, showing engagement of the drug for their specific target to decrease de novo lipogenesis. The question is this a relevant target for treating NASH? And that was in the second part of the study, or an expanded view that came after this initial part to look at target engagement. Does this actually have an effect in NASH patients? So taking this to the next phase of clinical development, they looked at 126 patients and treated these patients with this drug. There's 126 patients that have various degrees of NASH. So NASH is a spectrum of disease, of course, that is characterized by both liver fat as well as liver stiffness. So 2 imaging metrics were used to look at response to therapy. And there were 2 different therapeutic doses that were used, a 20 and a 5 therapeutic dose. And this is a liver-targeted oral delivery of this drug. And what they observed is that in the patients that responded, they saw sharp and profound decreases in, you only show 4 acetylcarnitines, but there is acylcarnitines, but there is a profound shift in acylcarnitines in the responder group at both doses that they saw. So that they actually developed a targeted panel of 22 acylcarnitines to be able to look at response to therapy from once they were able to assess that they were able to get biomarker target engagement. On all this showing good area under the curve for their responders. And this is important now because you can actually start to design rational trials. So you can understand who responds to a therapy, who doesn't respond to a therapy, stratify populations for efficacy of response, and now you're able to have data-informed rational design for clinical trials, larger clinical trials, to be able to understand the response, safety and efficacy profiles, and really be able to drive clinical development in a rational and designed way. So that example is when you know the target and target engagement. What about for novel applications for identification of novel targets? There's an example of that here with ketogenic diet that is used to be an effective treatment for refractory epilepsy. So we're kind of covering the spectrum of all diseases from cardiovascular disease and multimorbidities liver disease, and now we're talking about more neurological disorders. You can see, again, metabolics and metabolism is relevant across all disease states because every disease requires changes in biochemical steps. So it was known, this is work done by Elaine Tsao at UCLA, and it was known that the ketogenic diet is useful or beneficial or effective treatment for refractory epilepsy, but the mechanism wasn't known, and they wanted more to develop more targeted, more effective therapies, more consistent therapies around this, and understand the mechanism of the ketogenic diet. And the investigators thought that there was a microbiome component in all this. So they worked with us on a discovery approach to characterize or profile stool samples and circulating plasma samples from preclinical model of refractory epilepsy. And they found, of course, that the ketogenic diet was beneficial in this model. And through the profiling of the gut and circulating metabolites, they found that there was a decrease in the responsive group, the group that responded to therapy with a ketogenic diet, in a specific set of metabolites called gamma-glutamyl amino acids that derive from a specific enzyme that drives the class of these molecules, that it decreases in the response group. And they knew there was a microbiome component, so they did antibiotic treatment to see if to test the power of that association. So they treated these animals with antibiotic to knock out the microbiome, and they lost their effect. And then they reintroduced a specific strain that was able to recapitulate the ketogenic diet effect. And all these correlate with decreased levels of stool and circulating metabolites of this class of molecules, gamma-glutamyl amino acids. And this ultimately results into a change in the brain, hippocampal slices, again profiling these, where the changes that are occurring in the gut and the circulation result in changes in the GABA-glutamyl glutamate ratio in the brain that associate with better outcomes in the ketogenic diet. So what this really suggests is that targeting the nodal regulator of the enzyme that makes this could be a good target. They've developed an inhibitor specifically to gamma-glutamyl amino acids, the enzyme that makes all this class of metabolites, and they saw a significant increase in the effect of their phenotype in the absence of any ketogenic diet. So they saw a direct inhibition from oral administration into animals of an inhibitor for gamma-glutamyl amino acids. And of course, they worked out the mechanism behind that, and so they identified a novel target, showed efficacy of inhibition in this model, and this formed the basis of a new biotherapeutic company that has continued to develop that specific inhibitor, as well as inhibitors using metabolic profiling across all different neurological disorders. And speaking of neurological disorders, the last one I'll talk about that again focuses on the microbiome is amyotrophic lateral sclerosis, which is a disease of motor neurons for which there are few treatment options. So again, a profound neurological disorder that patients don't have a lot of hope in terms of treatment regimen or treatment profiles. And this is work done by Ron Segal again at the Weizmann Institute, looking at and showing a translation model starting preclinical and then translating up to humans, that looking at superoxide dismutase transgenics is a model, a preclinical model for ALS. And they knew there was a microbiome component to this. So there's a lot of work to get to this figure here, because they did all kinds of serum-free, serum pathogen-free mice, germ-free mice, and antibiotic treatment to know that the loss of gut complexity or diversity was impactful, that it resulted in worse outcomes in this animal model. So they knew they were losing some sort of beneficial strain. They did a lot of work to characterize that strain from the metagenomics, and they found a specific strain called Akkermansia muciniphila that appeared to be beneficial for promoting healthful outcomes or beneficial outcomes, improving the phenotype of their model. And that's shown here. So you can see in the relative to control mice that show no effect, obviously, for ALS, that the ALS mice here, in the absence, so these are treated with antibiotic, that they have no gut microbiome, and they have very strong phenotype for ALS, and that when you gut implant or re-inoculate a specific strain into their gut, you get a massive significant improvement in the profile or phenotype of these animals. So a ton of work to do, metagenomic profiling that gave them the strains that were present and differentially present that could potentially be involved, only told them what the strains were, didn't tell them anything about what those strains were producing. So what was the functional output? What is the mechanism of action for how the gut microbiome, what its outputs are that influence health outcomes in this model system? So we worked with them at this point to take essentially this group right here, control animals, and then the ALS animals either with vehicle treatment or the Akkermansia muciniphila inoculation, to profile those using global discovery platform to understand what they're producing. And lo and behold, a massive signal appeared for nicotinamide in the Akkermansia treated animals that associate with good outcomes. And there were some other signals that emerged too that they tested, but obviously this is the paper and this is what they talked about. So nicotinamide and all the many metabolites within the nicotinamide pathway were significantly increased, showing that the entire pathway was upregulated in these animals that had been inoculated with this specific strain, leading to the question or hypothesis that this could potentially be involved, the beneficial agent that mediates this beneficial effect. Well, they tested that and looking at animals dosed specifically with nicotinamide versus placebo, significant and massive improvement in their phenotype, showing that nicotinamide alone was able to, presumably derived specifically from the super-producing strain of Akkermansia, was able to drive beneficial outcomes in this model system. And as a translatability question, individuals, humans, both in CSF and in serum, were able to show decreased levels in CSF and circulation of nicotinamide in ALS, kind of consistent with the story that at least to improve treatments or improve outcomes for these patients who really don't have any recourse, that this is one potential avenue for a target identified and potential treatment that came from a global untargeted discovery platform. So just to summarize, kind of pull everything together, metabolomics is all about providing unparalleled coverage across as many biochemical pathways to provide as much coverage as possible. And in terms of the human health cohort space, this comes from study powers as well. And I think metabolomics, hopefully you've gleaned that Metabolon is able to do this at scale and have the coverage and capacity to really provide biological insights. Our technology has been instrumental across a number of different disease areas. Arguably, I would say one of the most innovative is to apply this in a multi-omics kind of way to functionally map genes of unknown function, variants of unknown significance, largely in genes of unknown function. And we've actually been successful using this approach, or our technology has been successful in using this approach to functionally map over 800 genetic variants, the function of which were not known. And when you think about our function of all the genes that are out there, adding 800 to the mix is a huge win and massive add to the knowledge of the field. And that biochemically profiling, because as Brian said, our microbes message with us through small molecule signaling. Biochemical profiling is uniquely suited to be able to elucidate those signals, deconvolute those signals, and help understand their role in influencing human health outcomes. And because every disease requires a change in a metabolic step or reversion from disease back to a homeostatic step, metabolomics really represents a powerful technology and a cornerstone molecular phenotyping tool. So with that, I know we covered a lot of grounds, or a lot of case studies we went over. I'll stop there and happy to address any other questions, and Simone and Brian can also weigh in as well. So thank you for your time. Real pleasure to be here.